Hey everybody, how's it going? Today we're going to look at DNA replication, which is the process by which DNA is copied within a cell. This video will go along with the 12-2 review worksheet posted in the classroom. Let's get to it. Semi-conservative DNA replication is a method of DNA replication in which the original strands of DNA, called the parental strands, separate. They then serve as templates to produce two new DNA molecules, each of which have one parental strand and one new strand. This can be accomplished thanks largely to the complementary nature of DNA. Remember from the last video, we said the DNA strands are complementary, or that the nitrogenous bases of one strand of DNA will match up with those of the other. But we'll come back to that in just a moment, as there are a few things that need to happen before we can even think about copying our DNA strands. As we said last time, DNA spends most of its time in the cell packed very tightly. DNA wraps around histones to form nucleosomes, which coil to form chromatin fibers, which coil further to form chromosomes. In order for DNA to be replicated, it must be unpacked and unzipped. An enzyme called helicase is responsible for unwinding the DNA. Another enzyme, topoisomerase, keeps the DNA from reforming its coils. Helicase can then perform its second function of unzipping the DNA. This means that helicase separates the two strands of DNA by breaking the hydrogen bonds in between base pairs, resulting in two strands. Wherever helicase begins unzipping the DNA is the first part that will be available to be copied, and this is called the origin of replication. Wherever helicase is unzipping the DNA molecule, the strands separate from each other, forming what is called a replication fork. In order to prevent the strands from coming back together, single-strand binding proteins bind to each separated strand. Now that we have our two parental strands of DNA separated, we're ready to start making copies. The first step is carried out by an enzyme called RNA primase, which adds short segments of RNA, called RNA primers, near the origin of replication. These primers allow the enzyme DNA polymerase to begin forming our new strand of DNA by adding the appropriate nucleotides at the 3' end of the new DNA strand. Crucially, DNA polymerase can only add nucleotides to the 3' end, which means that new DNA is always formed in the 5' to 3' direction. For the parental strand oriented 3' to 5', the one shown on top here, this isn't an issue, as its complementary strand would be oriented correctly. We call this the leading strand, and replication here is said to happen continuously, as DNA polymerase can synthesize new DNA as the DNA unzips, never running into any issues. The same cannot be said for our other parental strand, oriented from 5' to 3' and shown here on the bottom. We call this strand the lagging strand. Because its complementary DNA strand will be oriented 3' to 5', DNA polymerase cannot simply follow the helicase and continuously synthesize new DNA. Instead, RNA primers must be added along multiple points as more DNA is exposed, and multiple DNA polymerase enzymes must bind and add new DNA in short segments. These short segments of DNA are known as Okazaki fragments. When replication is completed, the RNA primers on both the leading and lagging strands must be replaced with DNA, and the Okazaki fragments on the lagging strand must be linked together. DNA ligase is the enzyme responsible for sealing gaps in the new DNA strands, resulting in two new DNA molecules. Each cell in your body has about 3 billion base pairs that need to be copied each time a cell divides, which all occurs in about an hour in a typical human cell. This might seem like a recipe for a large number of mistakes, but eukaryotic DNA replication is actually very accurate, and it has to be, as small mutations or mistakes can result in non-functional cells or even cancers. DNA polymerase is highly accurate, but it also performs a proofreading function that allows it to catch its own mistakes. The reason that DNA replication can be so accurate is thanks in part due to the complementary nature of DNA strands. The parental strands are used as a template for the new strands, and DNA polymerase adds nucleotides such that the bases are complementary. Remember from last time we said that adenine will pair with thymine and cytosine will pair with guanine. Let's look at how this allows us to copy DNA accurately. Here we're given 3 template strands of DNA. The first two are oriented 3' to 5', and the bottom one is oriented 5' to 3'. That means these first two, here and here, are going to be leading strands. This one's going to be a lagging strand. Now, because of the complementary nature of DNA and the knowledge that DNA strands are always going to be running anti-parallel to each other, we can start filling in the gaps on what the new DNA strand would look like synthesized from these template strands. For this first one, since it's oriented 5' to 3', we know our other strand, the new one, is going to be synthesized or is going to be oriented 5' to 3'. So we can draw in the 5' on that side, the 3' on that side. Then all we need to do is make sure that these bases are paired with the opposite base that they go with. Adenine will always bond to thymine, so here we'll put a T. Thymine will always bond to adenine. Cytosine will always bond to guanine, and guanine will always bond to cytosine. So in that way, you can fill out the entire strand. And then you've done. You've got your new DNA strand that is complementary to your top one. Now, take a second, pause the video, and give the second one a shot before I go through how to do it. So, our template strand is once again oriented 3' to 5', so we know that our new DNA strand is going to be 5' to 3'. Then all we have to do is match the bases, so T goes to A goes to T, A, T, C goes to G goes to C, and so on and so forth, until we've got our new DNA molecule completely synthesized. This one last practice has a little bit of a different orientation, so there's a small difference right at the beginning, but go ahead and take a moment and see if you can fill that one out as well before I go over it. Right, so the main difference here is that this last strand is oriented 5' to 3', which means that our new DNA strand is going to be 3' to 5'. And then after that, it's all the same thing. So A to T, C to G, T to A to T, etc., until you finish this one out. And it should, when you're done, look like this. The last thing we want to note is the differences of DNA replication in eukaryotes and prokaryotes. Eukaryotic cells can have multiple origins of replication, while prokaryotic cells, which have circular DNA, can only have one. DNA replication in eukaryotes occurs in the nucleus, while prokaryotes, which lack a nucleus, replicate their DNA in the cytoplasm. Now let's take a quick moment to review. Before replicating, DNA must be unwound and unzipped in order to be accessible for replication. Helicase unwinds the DNA, then unzips it at the origin of replication. Topoisomerase prevents the DNA from reforming coils, and single-strand binding proteins prevent the separated strands of DNA from coming back together. This forms a replication fork, with a leading strand that can be synthesized continuously and a lagging strand that must be synthesized discontinuously by the formation of Okazaki fragments. RNA primers, placed by RNA primase, start the replication process and allow DNA polymerase, a highly accurate enzyme, to add nucleotides to the new DNA strand. New DNA is synthesized in the 5' to 3' direction. After replication is complete, RNA primers are removed and replaced with DNA, and the enzyme DNA ligase seals the gaps and results in the formation of 2 new DNA molecules, each with 1 parental strand and 1 new strand. This is called semi-conservative replication. That's it for this video. Be sure to check out the other DNA replication video posted on the classroom, and I'll see you next time.